So these notes are going to be about one of the most amazing things in all of biology, and that is how cells can use the code of DNA to make proteins, which is the #1 job of a cell because we're all made of proteins and proteins carry out all of our chemical reactions. So this is just absolutely incredible to me how this works, that it's so simple and easy and also kind of complicated. So all of those things together, I know it sounds contradictory, but they're all true, and it's just really an amazing process. So I hope you can follow along with me. We're not going to do the whole PowerPoint. We're going to break it up into chunks, but I'll try to get through both transcription and translation in this segment. So first of all, after Watson and Crick proposed the structure for DNA, the first question immediately that needed to be answered was, okay, that makes sense, but how does the DNA molecule actually serve as a code for proteins? And of course, the first thing that people suspected was somehow the combinations of bases must code for specific amino acids. As you know, there are 4 bases: adenine, guanine, cytosine, and thymine. And so the first thing that came up was, well, gee, how many different combinations can we make of 2s of those bases? Because clearly you can't have a scenario where one base stands for one amino acid, because how many amino acids are there? Well, there's 20 amino acids, and there's only 4 bases. So it can't be one base equals one amino acid, but how many different combinations of 2s can you make when you have 4 different things? And those of you who have done this recently in math will know that it's 4 to the 2, or 4 squared, and that equals 16. So 16 different codes is still not enough to code for the 20 amino acids that make up proteins, because again, we need to code for all 20 of them to make all the proteins that are present in living organisms. So they didn't stop there. They just said, well, let's just go on and try the next one. So how about different combinations of 3s? Well, there are 4 bases, so we can arrange them into 64 different combinations of 3s. And clearly this would be enough different codes to code for the 20 different amino acids, and in fact it seems like way more than enough, but as we'll see, it's kind of a good thing that there are some extra ways to code for the different amino acids. So scientists immediately then decided, well, how can we test out this suggestion or hypothesis that it's groups of 3 bases at a time that code for each one amino acid? And, since they were so clever, they quickly figured out that they could automatically synthesize some pieces of DNA consisting of known sequences and put them into test tubes with some necessary enzymes and some nucleotides and proteins and just all the stuff that a cell needs, and see what happens to that DNA, see if it gets turned into any specific proteins, if any proteins appear in the test tubes after a while. And then they analyzed those polypeptide chains or proteins that did appear. And in fact, this was very interesting. For example, if they synthesized a piece of DNA that just consisted of repeating CAC, ACA, CAC, ACA, and so forth like this, they hypothesized that depending on where the ribosomes, where it was known proteins were made, depending on where the ribosomes started to read this sequence, you would either get proteins made of the same 2 amino acids over and over again, if it was groups of 3s, because you would have CAC coding for one amino acid and then ACA coding for another, or you would get groups of, or strings of proteins made only of one amino acid, or the same one over and over again if it was groups of 4 or something different. So for example, groups of 4 are going to give us CACA, 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 and so forth. And what they found was when they looked at the resulting proteins that got built in the test tube, they found that it was always 2 over and over again. So when they alternated 2 letters like that, they got great big long polypeptide chains made of 2 different amino acids. And doing this, they could even figure out what the code was for all of the 20 different amino acids. So this is how we figured out that ACA is going to code for valine and CAC codes for cysteine. So that was a pretty smart experiment. And there is some redundancy in the code, quite a lot in fact, and what that means is there are different ways to code for valine. There's not just one way to do it, not just CAC, and I'll teach you about that in a little bit. There's also some triplets, that is groups of 3 bases, that mean stop, or that tell the ribosome this protein is finished. Now this is an example of the codon table that scientists were able to work out in the 1950s as a result of all of these experiments. And I'll show you how to read it in a little bit, but for now I just want to show you that we've got it all worked out and we know exactly what string of amino acids will be coded for by any particular sequence of DNA, and it's not rocket science, it's not difficult, and you can easily look it up and figure it out yourself. So given a string of bases, you can figure out what is the amino acid chain that's going to be formed from that if you put it into a cell with some ribosomes and some amino acids. So as you can see, there are many different ways to make serine, many different ways to make proline, glycine, and so forth. You have a lot of different codes for, in fact, for making valine and so forth. I'll show you again in a minute. So the first step in the process for a cell to make a protein out of its DNA is called transcription. We're going to have 2 steps, transcription and translation. The first step is called transcription. In transcription, the cell solves a problem, the problem being that the instructions for the protein in the form of the order of the bases is contained only in the nucleus in the DNA. And the DNA has to stay in the nucleus because it's much too big to get out and too critical to the functioning of the cell and the organism to allow it to be unprotected. So it's inside the nucleus, which actually has a double membrane, and it stays in there, kind of like locked up in a vault. And the proteins are synthesized in the cytoplasm at the ribosomes, as you should remember, and those are outside the nucleus. So somehow the message has to get from inside the nucleus on the DNA to outside where the ribosomes are. And therefore, the message is copied, and that word to copy it is called transcription, or to transcribe something. The message is copied and then carried out to the cytoplasm. Here is just a reminder of all of the different organelles that are in a eukaryotic cell, this being an animal cell. And the big purple blob here, that's the nucleus, and notice there are holes in the outer envelope of it, and those are called the nuclear pores. So the message is going to be copied and then carried out through those little holes to one of these little blue dots. These little blue dots in the cytoplasm, those are the ribosomes. So in order to understand how transcription happens, you have to know what RNA is. RNA is another nucleic acid, very, very similar to DNA, but 2 things are different. It contains ribose instead of deoxyribose for its sugar. It's exactly the same shape, a 5-carbon sugar, just like deoxyribose. But as you can imagine, it has an extra oxygen, so deoxyribose is missing that one oxygen that ribose has. And then it has a phosphate group on it, just like a DNA nucleotide has, so I'll draw that there. And then it has a base sticking out, attached to the sugar, to the number 1 carbon on the sugar. And the 4 bases are almost the same as they are in DNA. We have A, C, G, and they match up with their same partners just like they do in DNA. But then the problem is there is no T in RNA. So instead of T, we have a base in RNA called uracil, which we represent with a U. Now, on the one hand, this is kind of confusing and messy, and if I haven't mentioned it already, I want you to know that life evolved as a series of mutations, and that's why it's so messy and complicated. That's why it sometimes seems to us to be like a Rube Goldberg device doing things in a crazy, messy way. And it doesn't really make sense that RNA should have a U instead of a T, but the other bases being the same. And it's just a little glitch and something that you're going to have to deal with and learn about. On the other hand, it makes it easy to know when you're looking at a string of letters whether you're looking at RNA or DNA. If there's U's, it's RNA, and if there's T's, it's DNA. So the first thing that happens is the DNA that contains the needed gene, that is the recipe for the protein that the cell needs at the moment, unzips down the middle just like it unzips when it's going to replicate itself. And then these RNA nucleotides that are floating around in the nucleus go and line up, match up with the opposite bases on the DNA, and form a chain or a strip of messenger RNA. So this single-stranded copy in opposite bases, so not an exact copy of the gene, but the opposite base copy of the gene is made, and we call that strip of nucleotides messenger RNA. That's what the little M stands for. And of course there's the main enzyme carrying out that job, and it's called RNA polymerase. So I just want to draw this process out for you a little bit. Let's say this box here is the nucleus, and here is a piece of DNA in our nucleus. And let's say here's the gene for the particular protein that the cell needs. So that DNA is going to unwind right there and expose the bases that make it up. I'm just going to make 6 of them. And to make it easy, I'm just going to make it C, 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 A, A. Okay? So that's the gene. Now there is no gene that's really just 6 bases long, but just to demonstrate it for you, we'll make it easy like that. And of course the opposite bases are over here, and we're not going to pay any attention to those. Those are not the particular gene. Later on down on this strand of DNA, the gene might be on this side. So it could be on either side, but right now we're going to look at this section. This is our gene. The next thing that happens is the RNA nucleotides that are floating around here in the cytoplasm or of the nucleus are going to go and line up. So this is my rendition of a nucleotide. They're going to go and line up in the correct order by matching themselves with opposite bases. So if this is G, 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 then those can fit here, here, and here. And all these little nucleotides will come and line up there and create a strip of messenger RNA. So we'll add the other 3 that we need. Of course, what matches with A in DNA is T, but we're making RNA, so we're going to put a U instead of a T, and another U, and another U. So now this is our piece of messenger RNA that has been transcribed or copied from the DNA in the nucleus. And this piece of messenger RNA will go out through one of those holes called the nuclear pores out into the cytoplasm to a ribosome. And the ribosomes are actually, if we look at them up close, kind of blobs that look sort of like that, made of a big subunit and a smaller piece. And so we have made our piece of messenger RNA, and it's going out into the cytoplasm, and that process is called transcription. Step 1 in making a protein. Now, if you've gotten all the notes and you understood everything here, you can go on and watch the next segment of the video, which is called translation. If you need to, though, go back and watch this one again.